Hello friends, how are you? Welcome back. In the previous video, we have talked about initiation and elongation of transcription. Okay, in today's video, I will be talking about the termination. That is little bit complex process as compared to initiation and elongation. So before starting the termination, let me give you a brief summary of elongation. Okay, in the previous video, we have talked that in the initiation, the transcription initiation complex will be formed, and this transcription initiation complex consists of transcription factors and RNA polymerase, that is having carboxylic terminal domain or C terminal domain, and a very important transcription factor, that is transcription factor 2H. That will be having this transcription factor 2H will be having helicase activity, and it will be having kinase activity. With the help of its kinase activity, it will cause the phosphorylation of C terminal domain, and when this phosphorylation will take place, when the phosphorylation of C terminal domain will take place, there will be a conformational change in RNA polymerase enzyme, and because of this conformational change, all the transcription factor will detach other than transcription factor 2H and transcription factor 2F. All other transcription factor will detach, and then this RNA polymerase will start moving along the gene and start transcribing the gene. It will read this 3' to 5' end. It can only read 3 to 5'. So it means when it will move in this direction, transcription will take place at only one strand. It cannot read 5' to 3'. So it means at this strand, transcription will not take place. So the strand against which RNA will be formed is called as template strand, and the strand that is opposite to the template strand that remain intact is called as sense strand. Sense strand. So when the RNA polymerase will keep on moving, what will it do? Against C, it will add G. Initially it was like this. Against C, G was added, phosphodiester bond was made. Against C, G, C was added, hydrogen bond was made, phosphodiester bond was made. Against A, U was added, phosphodiester bond, hydrogen bond was made, and against T, A was added, and so on. So I explained in the previous video that the affinity between this DNA and RNA strand is not that much good as compared to this DNA and this DNA strand. So what will happen? These bond, hydrogen bond will be broken down. This RNA template will come out of the DNA, and the bond between the original DNA nucleotide will be made, and the DNA will remain intact. Similarly, transcription factor 2H, that is having helicase activity, it is responsible for the breaking of these hydrogen bond in the DNA strand. This transcription factor 2H, again it will keep on breaking these bonds, and new nucleotide against C, G will be added, against C, G will be added in this way, and later on this whole DNA strand will come out, and the bond between these DNA nucleotide will be formed again. In this way, the process of elongation will keep on going until the transcription ending point reaches. Like transcription initiation point, there also exists a transcription end point. Transcription end point. Well, we will discuss about the termination right now. Before talking about the termination, let me tell you one more thing. While moving, the RNA polymerase keep on moving for the transcription. there is a problem. We know that the DNA strand is not present as such. It is wrapped around the histone protein. It is wrapped around the histone protein. So what will happen if a histone protein come in the way of RNA polymerase? So it means now RNA polymerase will not be able to move forward. Now what will happen? Does the transcription terminate? No, it will not work like this. What will happen? There is a protein that is called as facilitate chromatin transcription. This protein will help RNA polymerase by removing the histone protein. These are the histone protein. Here you can see our DNA is wrapped around the histone protein, and these histone protein will be taken out by the FACT protein, and when RNA polymerase will move ahead and the transcription will take place, and the original DNA strands will again come back to the original position by attaching with each other. Now this FACT protein will again place the histone protein to its original position. Okay, so FACT protein is responsible for removing histone protein and placing back it to the original position once the RNA polymerase will move forward. So in this way, FACT protein help the RNA polymerase. Now let's talk about termination. Well, termination is little bit complex as far as RNA polymerase 2 is concerned. The genes that are being transcribed by RNA polymerase, they will be having special terminating proteins, and these terminating proteins will come at the ending point of transcription. They will attach, and RNA polymerase will not be able to move forward, and transcription will end. Similarly, for RNA polymerase 3, hairpin loop-like structure will be formed. That I have explained in that transcription in prokaryotes. Hairpin loop-like structure is formed. Exactly same mechanism will be followed. But as far as RNA polymerase 2 is concerned, transcription is little bit different and little bit complicated, and it is least understood right now. But what is the mechanism? Let's talk about. So RNA polymerase will keep on moving, and it will keep on adding, and it will keep on breaking these bonds, hydrogen bonds, with the help of transcription factor 2H, and it will keep on adding. Against T it will add A, against T it will add A, against T it will add A, against A U, against T A, against T A. In this way it will keep on adding, and in this way you can see A, UAA, and there are some other nucleotide, and then UG rich area, like there are lots of UGs, uracil and guanosine are present. Now why I am talking about AA, UAA and why I am talking UG rich area, let me tell you. Well, as far as RNA polymerase 2 is concerned, it will not stop at the transcription endpoint. It will keep on moving. It will keep moving downstream the endpoint. Approximately 1000 to 2000 nucleotide will move ahead of the endpoint. It will move ahead of the endpoint. Now how the transcription will stop, and who will stop the transcription? When this RNA will be formed, it will be transcribed. No matter this RNA polymerase, this is the RNA polymerase. This RNA polymerase is keep on moving forward. When this RNA will be formed, a region that is called as AAUAA and a UG rich area has been transcribed on the RNA. RNA having these nucleotides has been formed right now, and this RNA polymerase is keep on moving forward. RNA polymerase is keep on moving forward. Now what will happen? This sequence is recognized by this protein, CPSF protein, and this protein will come and attach over there, CPSF. Okay, let me write CPSF. Similarly, UG rich area will be recognized by CSTF protein and will be CSTF protein, and will attach on the UG rich area. Now what will happen? Listen to this point very attentively, please. RNA polymerase has not stopped working yet. It is keep on moving, but this RNA has been formed and the sequence has been recognized by these special proteins. Now what will happen? This CPSF protein will cleave down, will break down this phosphodiester bond, and this strand of RNA will be formed, and this is the original RNA strand that will be further processed by adding methylated cap at the 7-GM methylated cap at the 5 end and poly A tail at the 3 end. Okay, if this strand is being transcribed, that is from 3 to 5, obviously the RNA that is being made, it will obviously 5 prime to 3 prime. So this is the 5 prime and this is the 3 prime. So this 5 prime has been capped with the 7-GM in RNA processing, and with the help of poly A polymerase enzyme, some other adenosine nucleotide will be added and a poly A tail will be formed. Now what happened to the RNA polymerase? It is keep on transcribing. Now how to stop it? Our desired RNA has been formed and this is ready for the translation now. It will go, it will attach on the ribosome and start the process of translation. But what to do with this enzyme that is keep on moving, and it has moved away from the end point approximately 1000 to 2000 nucleotide away from the end point. There is a protein that is called as XRN2 protein. XRN2 protein. Now it will recognize this 5 prime RNA that is still attached with the RNA polymerase, and it will act as a 5-exonuclease. Now what is nuclease enzyme? Nuclease enzyme is an enzyme that has ability to break these phosphodiester bonds. Okay, you know that the bond between these two nucleotides are phosphodiester bond. These are phosphodiester bond, and these one strand and other strand, these bonds are basically hydrogen bond. So 5-exonuclease has the ability to break these phosphodiester bond. Now once this XRN2 protein has attached over there, it will keep on breaking these bonds. And all the RNA that has been formed, it will be dissolved. And now this RNA polymerase will detach from the DNA and DNA will stay intact. Now our newly formed messenger RNA has been formed, and this assembly has been broken down with the help of XRN2. So this is the process of termination in eukaryotic transcription. I hope you like my video. If you have any question, please do not hesitate to ask in the comment box, and don't forget to subscribe and like my video and channel. Thank you so much.